Sugars. We love to eat them, hate to learn them for the MCAT. In this video, you're going to learn the names, the structures, the isomer forms, and the bonding characteristics for all the types of sugars that you need to know for testing. We're going to start with monosaccharides. The 5 monosaccharides you'll need to know are glucose, mannose, galactose, fructose, and ribose. Then we'll move into the 3 disaccharides that you'll need to know for the MCAT, which are lactose, maltose, and sucrose. We'll learn the polysaccharides, glycogen, starches, and cellulose, and we'll also talk about what all the prefixes like D, L, alpha, beta mean, and how to define and identify reducing versus non-reducing sugars. So let's get started. All right, so we have here the sugars, the monosaccharides that you're going to need to know for test day. And we're going to start by looking at some common features, then we're going to go through each one. So I want you to look at sugars 1, 2, 3, 4, and 5 here. And what you'll notice is they all have 6 carbons, 12 hydrogens, and 6 oxygens. However, they all have different orientations, different connectivity between their functional groups, which means sugars 1 through 5 are all isomers of each other. So as we go through each sugar, we're also going to identify the isomer form that each of them are to each other. Because remember, if they have the same molecular formula but different connectivity, they are isomers. So let's start with sugars 1 and 2, and both sugars 1 and 2 are glucose. They're different forms of glucose. Glucose is our most commonly tested sugar on the MCAT because, of course, it's our main source of energy in our metabolic processes in our body. So I want you to note that glucose, along with the 2 other sugars here, have an aldehyde on carbon 1. That makes these guys all aldoses, okay? So anytime you have an aldehyde as a part of a sugar, they're an aldose. OSE suffix for sugar, aldehyde. All right, so we have carbon 1 having aldehydes on sugars 1, 2, 3, 4, and 6 here, which we'll talk about shortly. Now I want you to look at these. These are both glucoses, all right? They both have an orientation where we have 3 hydroxy groups on one side and 1 on the other. My mnemonic here is 3 and 1 for my glucoses, but they are different, right? If we look at them, each carbon, each chiral carbon, carbons 2 through 5, are exactly flipped. See how the OH is on the right here, left here, right? Hydrogen left and right here. So these guys are exactly flipped. In other words, if we drew a mirror, right, image, and we flipped them on top of each other, they would line up perfectly. But if we superimposed them, if we laid sugar 1 on top of sugar 2, they would not line up, right? The substituents would not line up. So do you remember what type of isomer that is when they're non-superimposable mirror images? They are enantiomers, all right? So these guys are enantiomers of each other. And the way we define enantiomers in sugars is we look at carbon 5. So if we look at carbon 5 here, what we look at is where the hydroxy group is on carbon 5 in a Fischer projection. If it is on the right, it is D. We label it D-glucose. And if it is on the left, we label it L-glucose. So D and L forms of the same sugar, D-glucose versus L-glucose, D-galactose versus L-galactose, are always going to be enantiomers of each other. So we have here the D form and the L form of the sugars. Now for you to know, all of our sugars in our human bodies are always going to be D sugars. All right, so typically speaking, if it's in the human body, if you have to guess, it's going to be a D sugar, but now we know how to identify it. All right, and that is glucose. Now let's move on to our next sugar, which is in the center here. And this one is mannose. And mannose has 2 of the same on the top and 2 of the same on the bottom. So 2 hydroxys on one side and then 2 on the other. So 2 and then 2. And that's how we got mannose. All right, mannose, M-A-N-N-O-S-E is our next sugar. Now you'll notice that we still have an aldehyde, so we're still an aldose. And on this one, mannose has the OH on the right, so this guy is going to be D-mannose. Now I want you to compare D-mannose to D-glucose for a moment. What is the difference? You can pause this video. What is the difference between D-glucose and D-mannose in terms of their structure? Go ahead, pause the video and try to identify. All right, so if you identified that carbon 2 was the difference, you are totally correct. So you'll notice here that glucose D-glucose and D-mannose have exactly the same structure, except at carbon 2, our OH is flipped. Now, we don't worry about the orientation of the aldehyde because it's not a chiral carbon, right? A double bonded carbon is not chiral. But carbon 2 is a chiral carbon and it is flipped. OH is on the left for D-mannose and on the right for D-glucose. So here is one of our cool rules. If we have multiple chiral centers, but we only differ at one of them, we are known as an epimer, all right? An epimer. So it is specifically a C2 epimer of D-glucose is what we call it. All right, C2 epimer of D-glucose is mannose. All right, so epimers are diastereomers. They have multiple chiral centers, but they only differ at one of them. All right, if we were comparing D-mannose to L-glucose, we've got multiple differences. So these guys are just diastereomers of each other. Diastereomers are when we, diastereomers, all right, or when we have multiple chiral centers and multiple differences, but they are not the mirror images, right? They're not mirror images of each other. So enantiomers, they have to be mirror images. They have to exactly mirror each other. Diastereomers, they just have multiple differences, but they don't line up as mirrors. And epimers have only one difference between them, even though they have multiple chiral centers. Cool. Let's do this again with our next sugar. So our next sugar here is known as galactose. Galactose. All right, is our next monosaccharide. Again, it's D-galactose. Again, our bodies use D sugars, so preferentially I usually draw our sugars in their D form because that's the most common. And in here we can see a 1 to 2 to 1, galactose, 1 to 2 to 1 ratio of where our hydroxides are. So again, I'd like you to pause this video and compare D-galactose to D-glucose and D-mannose and see the differences between each. So go ahead and pause the video, see if you can find the differences between each. Okay, so for D-galactose and D-glucose, we can see that the only difference is at carbon 4. All right, so carbon 4, it's going to be a C4 epimer, of D-glucose, D-glu, we'll just write, all right? But if we look at mannose and galactose, right, they're different. They have a difference at both C2 and C4. So these guys are also diastereomers. I'm just going to write dia of each other. All right. So mannose and galactose are epimers of glucose, but diastereomers of each other. Cool. So this is a great way to review isomers with sugar since they're all isomers of each other. Now let's go down to our second row, and I want us to look at how these guys are very different. So let's look at sugar 5 first. What's the big difference in sugar 5? Well, it's got this ketone group instead of an aldehyde. That makes this guy a ketose instead of an aldose because it has an entirely different functional group. It's not just different orientations around a carbon, it's a totally different functional group. It has a different IUPAC name. That means that this sugar is what we call a structural isomer, also known as constitutional, structural isomer of sugars 1 through 4. So these are structural isomers, which means they have a totally different functional group, different name, right? It's not just different connectivity, but they still have the same molecular formula. So more different, still molecular formula. Bonus points if you know what this sugar is. This is fructose, all right? So fructose is kind of our odd man out. It's our special guy where it's still a 6-carbon sugar, but it's got a ketone on carbon 2 instead of an aldehyde on carbon 1, all right? It is still D-fructose. We still have the OH here on carbon 5, so we still have D-fructose as well. Now our final sugar, sugar 6 here, is even more different than fructose because if we count 1, 2, 3, 4, 5, it's only got 5 carbons, which means it's not an isomer form of the rest of these sugars. It's still an aldose, but it's a 5-carbon aldose. And this guy in particular is known as ribose. Now, ribose is really important, not for carbohydrate metabolism, but because it's an essential structure of nucleotides. You might recognize the term DNA, deoxyribonucleic acid. All right, ribo meaning ribose. All right, RNA, ribonucleic acid. The ribo stands for the ribose sugar. Now, deoxy just means that in DNA, we don't have a hydroxy group on carbon 2, we just have a hydrogen. In RNA, they have the hydroxy groups on all 3 carbons here. All right, those were our 5 major monosaccharides you're gonna see on the MCAT, but there's a wrinkle. Usually we actually see sugars in their cyclic form in our body, not their linear. So we're gonna need to be able to recognize and work with them in their cyclic forms, which is what we're gonna talk about next. Before we do that, I'm Amanda Brem. I've been coaching students on their MCAT journeys since 2019, so please subscribe to this channel if you want more videos on learning MCAT content, test-taking strategies, and mental fitness tips to perform your best on test day. And if you'd like to join my next cohort of classes, please check out the link in the caption below to register. All right, so here are our cyclic forms of our sugar. I just have a few sugars up here because they all follow the same pattern. On the far left, we have what's called a Haworth projection. A Haworth projection shows it in this nice, comfortable, circular format. And then over here we have the chair projection. The chair projection represents what it actually looks like in the body, but interestingly on the MCAT we tend to use Haworth more often, so that's what we're going to focus on today. To form this cyclic projection, what we've done is what's known as an intra meaning within, molecular nucleophilic attack. So what we've done is carbon 5's oxygen here, and that's why we care about it with L and D, is going to go ahead and do a nucleophilic attack on the carbonyl carbon up in the aldehyde, and it's going to result in a cyclic molecule. You don't need to know all the steps of it, but it is important to note that this oxygen here has become this ring oxygen here, and this carbon 6 is up here, carbon 6 here, and carbon 1 has become the carbon to the right of the ring oxygen. This carbon is incredibly important. It's known as the anomeric carbon. All right, and it's a site of isomerization. So in each sugar, the OH on the anomeric carbon can either face down, like here, or up, like here on the chair projection. If it is facing down, it is known as an alpha sugar. The alpha denotation only works when we're in the cyclic sugar form. All right, so we're not going to be able to identify alpha or beta on our linear form. It's only going to come up when we're in our cyclic form. And because this aldehyde is forming the OH, right, it can really form either the alpha or the beta. We don't know until it happens. So by facing downwards here, it is alpha. If it was up here, like up here on the Haworth, or up-ish, like it is here over on the chair, it is a beta sugar. So up is beta, down is alpha. A lot of people use, alpha kind of looks like a fish swim down, right? So that's our alpha little mnemonic here. So that is how we identify an alpha or beta sugar. In order to switch from alpha to beta, or back again, right? Back or forth, we do a reaction called mutarotation. Mutarotation. It's a type of isomerization reaction in which we form just the other isomeric form, all right? So if we're figuring out alpha or beta, we always have to go to carbon 1, which is the carbon to the right of the oxygen. Now to determine L or D in a cyclic formation here, we just go to carbon 6. If carbon 6 is facing up, like it is here, it is a D sugar. So this carbon's also facing up, so it is a D sugar. So anytime our carbon 6, I kind of call it the flag carbon, right? 'Cause poking up like a flag is up, that means that it is D. So this is known as alpha D glucose. How do I know it's glucose? Because carbon 2 is down, carbon 3, the oxygen is up, and carbon 4, the oxygen is down. So we have a down, up, down pattern, which matches our down, up, down pattern here, carbons 2 through 4. So the pattern still needs to match what's happening in the linear sugar. Now, if you want a few more details with how the cyclic formation happens, everything on the right, all the substituents on the right on a Fischer projection end up down on a Haworth projection. So we can see here that carbon 2's OH is on the right, so it's facing down. Carbon 3's hydrogen is on the right, so the hydrogen is facing down. And carbon 4's OH is on the right, it's facing down. Now, personally, if it was me, guys, I would just memorize what this looks like. There's not that many sugars, and it's just easier to memorize what they look like in their cyclic projection and in their Fischer projection. Put them on some flashcards, get comfortable with those memorizations. Now the chair projection is a little trickier, but again, down is kind of like the angle is down. It's just whether we call this axial or equatorial is specific to chair projections. If it's kind of flatter, it's equatorial. If it's more vertical, it's axial. But again, the chair projection is not as tested on the MCAT, so the important thing is just to be able to identify alpha or beta if it does show up. Finally, I just wanted to show you the cyclic projection over here of a different sugar. Note that in this sugar, this glucose here, we have 1, 2, 3, 4, 5, 6 atoms in the ring, and 5 of them are carbons. Over on this sugar, we have 1, 2, 3, 4, 5 atoms in the ring, and 4 of them are sugars, and our 2 carbons are still facing up. So we still have 6 carbons, but 2 of them are outside of the ring. This guy is fructose. All right, so fructose, again, is the odd man out because instead of attacking carbon 1 up here in the aldose, it's going to attack carbon 2 where the ketone is. So this is fructose, and it is D because carbon 6 is facing up, and it is alpha because the OH is facing down. So we still follow the same rules even though it looks slightly different. So this is alpha D fructose here. All right, the last thing you need to know about these cyclic monosaccharides is some naming conventions. So for the fructose, we have a 5-membered ring with an oxygen in it, and that is known as a furanose ring. This is an organic word to describe a sugar ring that has a 5-membered, a furan is a 5-membered ring with an oxygen, OSE, sugar. We can also call this a hemiketal, right? That's also an organic term, a hemiketal where we had a ketose that we turned into a cyclic structure. On the other side, with our aldose that turned into a 6-membered ring, a 6-membered ring is known as a pyranose. Again, os, sugar, pyran, a pyran ring is 1, 2, 3, 4, 5, 6-membered ring with an oxygen in it. It's also known as a hemiacetal for the aldehyde, acetal ring. All right, so pyranose, hemiacetal comes from an aldose. I remember that as the mnemonic of PA, so the P's and the A's go together, PA. And then on the other side, this is a little non-PC, but the F's and the K's go together. So, you know, F's and K's go together, and that's how I remember furanose, fructose, ketal, ketose. F's and K's go together there. All right, so again, these types of naming conventions do come up often on practice questions on the MCAT, which means they're likely to show up on your test day. All right, we now get to move on to disaccharides. Disaccharides, 2 monosaccharides click together with a glycosidic bond. Glyco, sugar, acidic bond, so clicking together. So let's go through the 3 disaccharides you'll need to know for test day. This first one here is lactose. All right, lactose will come up a lot with prokaryotes because they have what's known as a lac operon, right? That's activated by lactose. So we have a lot of biotechnology that's related to this sugar. Now, lactose is a combination of galactose and glucose. So we have a glucose molecule and a galactose molecule clicked together here. So what we can see here is that both of these anomeric carbons, the OHs are up. So that means these are beta sugars. They're beta D because our carbon 6 is up as well. But this would be known as a beta bond, all right, because it's the beta sugars, and we label it by the carbon number that we're on. So this is carbon 1 of this sugar here, and this is carbon 4 of this sugar here, of this glucose. So we call this a 1,4 because carbon 1, carbon 4, beta, because it's beta, glycosidic bond. And that is how we identify any type of sugar bond, is we look at the 2 carbons that it's bound to, what's their number on the molecule, and then is it alpha or beta. So this is 1,4 beta glycosidic bonds connecting this lactose molecule together. This next one down here is maltose, all right? Maltose. Maltose, I like to think about like malt milkshakes, like extra sugary. These are just 2 glucoses, 2 glucose molecules clicked together. And we can see here that these guys both have their anomeric oxygens facing downwards, right? We still have D sugars, but now we have our anomeric oxygens facing downwards. So this will be an alpha 1,4 glycosidic bond. I should mention typically the alpha and beta go first. So glycosidic bond, and we call this alpha 1,4, we would call this beta 1,4 up here with lactose. All right, so this is an alpha 1,4 glycosidic bond. Up here, we have our special sugar out of the disaccharides, and this guy is sucrose. All right, sucrose is known as table sugar, right? It's our most common, like if you're just adding sugar to a cake, it'll probably be sucrose. And what we have here is glucose plus fructose. All right, and I want you to notice here, right, that we've got a kind of different situation where both of these other disaccharides, we have a free hydroxy group, right, that's nice and easy to connect and create more glycosidic bonds. But on this one here, we just have the CH2OH, right, on carbon 1 of our fructose. So the important thing to note here with our sucrose molecule is we still have a 1,4 bond, right? 1,4 bond. But in this case, we no longer have a free hydroxy group bound to our next anomeric carbon, right? So like this carbon 1 can't freely go ahead and just bind to other things. That means it is what's known as a non-reducing sugar. All right, reducing sugar, because often when we are doing reactions with sugars, it's oxidation reactions. In glycolysis, for example, we are oxidizing our sugars. We're removing electrons from our sugars to harvest them for the electron transport chain. So these guys are reducing sugars because, like a reducing agent, reducing sugars get oxidized, right? They reduce something else and they themselves lose their electrons or their hydrogens. So the interesting thing about sucrose is it cannot get oxidized further in the form it's in. So it is what's known as a non-reducing sugar. The rest of them, all the rest of these sugars we've been talking about, are reducing sugars. All right, so you just need to learn that for test day. This is another one of those secret rules on the MCAT. All right, this is a secret rule on the MCAT that sucrose is our only non-reducing sugar. So they're the odd man out. If they ask you which of the following is not a reducing sugar, we pick sucrose. There are two tests that test for reducing sugar that may be tested on the MCAT, and those are known as the Tollen's test and the Benedict's test. Both of them do redox reactions with metals. Tollen's uses silver and Benedict's uses copper to see if the sugar will reduce the ionized form of this metal. So if the sugar is a reducing sugar, in the Tollen's test, that silver ion will turn to silver solid, or what's known as a silver mirror, right? And in Benedict's test, if it's a reducing sugar, that sugar will reduce the copper and the copper solid will be precipitated out, all right? If you need a quick review on redox reactions, we do have videos on galvanic and electrolytic cells going through redox reactions basics. But for the purposes of our sugars, just know that sucrose is going to have a negative Tollen's and Benedict's test. And the rest of these guys will have positive tests because they're reducing sugars. All right, so that's just one of those little details that weirdly comes up a lot in the MCAT, and it's a good secret rule to know. Sucrose is your non-reducing sugar. To wrap things up, we're talking about polysaccharides. So polysaccharides are many, many individual monosaccharides clicked together. And of course, our major polysaccharide that we have in our body is glycogen. And that's our storage form of sugar. So this is glycogen here. This is what's represented, right? Glycogenesis is the formation of these glycogen bonds, which we talk about in our metabolism videos. Now, the important thing for organic chemistry here is to know what types of bonds are being produced, and there's two types. So if we look here at these chains, right, that's these straight chains of sugars, we can see we label 1, 2, 3, 4, and then 1, we can see these are our 1,4 sugars. And again, because the oxygen is facing down, it's alpha. So the main chain of glycogen is going to be alpha 1,4 glycosidic bonds, all right? Again, in our body, in our human bodies, we use alpha D sugars. Those are the sugars that we use to form glycogen and to break down in glycolysis and sugars. All of our enzymes related to metabolism, carbohydrate metabolism, are oriented for alpha D sugars. They will not work with any other isomer form. All right, so we have to convert if we need to use those. So our main chain here are our alpha 1,4 glycosidic bonds. And to do a quick connection to metabolism, our glycogen synthase, right, what makes our glycogen synthase, makes these bonds, makes these guys, all right? And to break them, all right, our glycogen phosphorylase, our enzyme breaks these bonds when we need to break down to our individual glucose sugars. Now you'll notice that these are all glucoses, okay? So these are just lots and lots of glucoses clicked together. But we have one more type of bond to talk about, and that's the bond here. All right, and notice here we still have it's at carbon 1 up here, but it's carbon 6 down here, all right? And this is kind of creating this branching, right? So these are known as alpha, it's still down, so it's still alpha 1, 6, because now we're on our 6th carbon here, glycosidic bonds, and these are known as our branching bonds, all right, our branches of glycogen. And the branches are produced by, it's a nice easy enzyme name, branching enzyme. All right, so branching enzyme makes these guys. Branching enzyme, we'll write down here. And debranching enzyme breaks them. Right, nice and simple. Makes and debranching breaks. Now, these branches are unique to glycogen. All right, so glycogen only are these alpha 1,6 branches. If we're talking about other starches, so other carbohydrate polysaccharides like starches will just have alpha 1,4 bonds, okay? So our starches will just be straight chain. All right, so I'll just write that up here for you. So starches are alpha 1,4 only. All right, and then our final polysaccharide that might be tested, so it's good to know, are our cellulose bonds. So cellulose, we cannot break down. Do you know why? There are sugars in plants and they are beta bonds. So they're beta 1,4 only. They're straight chains of beta connections. And again, they're still all glucoses, right? Everybody's polymers of glucoses, but they're clicked together with beta bonds instead of alpha bonds. And that's why we can't digest and absorb fiber, right? It's gonna go straight through our GI tract. We're not gonna absorb it because fiber is made of cellulose, which is made of beta bonds, which we can't do much about, right? We, with our human enzymes, cannot break these beta 1,4 bonds. It's also a cool little tidbit to know that beta 1,4 bonds are much stronger than alpha bonds. Alpha bonds, you can kind of see they're like pointed. They have a little bit of strain on them, so they're not very strong compared to our nice linear beta bonds. And that's actually valuable because our point of glycogen is to be able to break down to glucose quickly if we need the energy, right? So we want our alpha bonds to be easily breakable. Whereas cellulose, our sugars in plants, is used for structure, right? The plants use the cellulose for structure. They want those to be nice and strong. So it makes sense why beta bonds would be stronger and why we can't digest them because we don't have the enzymes for that. We do have some bacteria in our gut that break down the fibers into smaller chunks, but generally speaking, we're not getting our sugars, our glucose for fuel via cellulose. All right, and that was sugar structures that you may encounter on the MCAT. If you found this video helpful, please share it with your pre-med community. Remember that studying for the MCAT is hard for everyone and we can use all the help we can get. Hope that was helpful for you, and as always, happy studying.